we are Group 2, and my name is Chase Barker.
My name's Eduardo Ornelas.
I'm Abigail Chiu.
And I'm Manar Mohsen. To introduce the protein we've chosen, myoglobin is a low molecular weight globular heme protein found in vertebra muscle tissue. It requires 3 properties to function. First, a binding affinity to allow storage and transfer. Second, a low rate of auto-oxidation. And lastly, a low binding affinity of carbon monoxide to oxygen. We've chosen 1 MOB and 1 MOC, which are made of 154 amino acid residues and are commonly found in sperm whales. These chosen structures come with no mutations. A little bit about the functions of myoglobin include increasing aerobic capacity of cells through the storage of oxygen and facilitating the delivery of oxygen to the mitochondria. The information about these structures was obtained by X-ray. Myoglobin includes the following binding sites: 2 SO4 binding sites located at positions 52 and 53, 8 heme binding sites located at positions 40, 43, 44, 68, 72, 93, 94, 98, 100, and 104. And it also contains 2 metal binding sites at positions 65 and 94. 1 MOB contains a sulfate ion, water, and protoferrin containing iron, while 1 MOC contains the same except with added carbon monoxide.
From the total of 154 residues, there are 34 positively charged residues made of 4 arginine, 11 histidine, and 19 lysine. There are 21 negatively charged residues made of 6 aspartic acid and 15 glutamic acid. There are 18 polar residues made of 6 serine, 5 threonine, 2 asparagine, 5 gluten, and 5 glutamine. There are 66 hydrophobic residues made of 17 alanine, 8 valine, 9 isoleucine, 18 leucine, 3 methionine, and 6 phenylalanine, 3 tyrosine, and 2 tryptophan. There are also 15 special cases residues made of 11 glycine and 4 proline. We compared the 1 MOB sequence to the modeled sequence in the protein databank. We found that at position 64, there's glycine instead of histidine. At position 122, there's asparagine instead of aspartic acid. And both of these differences are shown in the top row here. Then we compared the 1 MOC sequence to the modeled sequence. At position 64, we found that there's threonine instead of histidine. At position 122, there's asparagine instead of aspartic acid. These differences are shown in the bottom row here. Then we made a superimposed, then we superimposed the 2 structures. The cyan is 1 MOB, the yellow is 1 MOC. We chose this structure because we thought this protein was interesting. It only has alpha helices, and the heme protein is also very cool. Comparing the myoglobin to hemoglobin, myoglobin is a monomer, whereas hemoglobin is a heterotetramer. Myoglobin only has 1 subunit and 1 heme group. Hemoglobin has 4 subunits and 4 heme groups.
Okay, so one of the cool things about BMD is that you can visualize the different types of residues. You can basically visualize anything you want. But anyway, so this is 1 MOB and all of the hydrophobic residues. And then in yellow, I have, this is 1 MOC, and these are also the hydrophobic residues. And then this is 1 MOB with the charged residues. So the red is the negatively charged residues, and the blue is the positively charged residues. And then we see the same here for 1 MOC. And then I decided to include all of the polar residues. So the additional green residues are only polar, they don't have a charge. And then we again see the blue and red residues. And then I have it here also for 1 MOC.
Okay, so next I will be showing you the difference between the heme in 1 MOB and 1 MOC. The one on the left shows the heme in 1-MOB, which you can see the iron in the center shows, the iron in the center is shown in pink, which has 4 nitrogens around it, bounded to it. So to the right, you see the heme in 1-MOC, which you can see the iron has a carbon monoxide molecule shown here. And so the iron in the heme binds to oxygen and acts as a transporter for oxygen. Since the 1-MOC has the carbon monoxide bound to it, this prevents the heme from being able to bind and transport oxygen. So here is I show the overlapping molecules. So 1-MOB is shown in orange and 1-MOC is shown in blue. As you can see, residue 152, which is glutamine, and 153 glycine right here on the left, are in different spatial arrangements, as well as the residues, as well as residue lysine, which is residue 96, is shown right here in the bottom middle. And then finally, residue arginine 45, which is arginine, is shown in the right. And so here's a zoomed in view of tyrosine 151, glutamine 152, and glycine 153. You can see there's a different spatial arrangement, especially seen for glycine right here. And then an interesting thing we found while playing around with the program is that the bond length between the alpha carbon in tyrosine 151 and glutamine 152 is 3.79 angstroms long. And so that's in 1-MOC, which is shown in blue. And while in 1-MOB, the bond lengths between the alpha carbon of tyrosine 151 and glutamine 152 is 3.82 angstroms, which is shown by the orange. So what we found is that MOB had a longer bond length than MOC.
Okay. The PDB files for 1-MOB and 1-MOC did include crystal water, which I have shown here, the sign being 1-MOB again, and yellow is 1-MOC. And something that surprised me about these results is that given the differences in the placement of the water molecules, I would expect more structural difference during the protein folding in a larger RMSD value. An RMSD value is a metric that quantitatively measures the similarity between 2 or more protein structures, and with an RMSD value of 0.552, which was evaluated from all of the 154 residues, shows that the protein structures are very similar. We saw in Eddie's visualizations that there were only 4 structural differences, and amongst those differences, the changes were very minuscule. As I'm sure you all know, typically the protein structures will form hydrogen bonds with the surrounding molecules and hydrogen bonds with potentially other molecules and its surroundings, which can affect how the protein folds and the structure it takes. And since the presence of these water molecules are so different from one another, I thought that the protein structure should have a much larger structural difference. This, therefore, is a great demonstration that myoglobin in general is a very rigid protein in order to efficiently reversibly bind to O2 and facilitate oxygen diffusion.